Hello everybody, and thank you for attending the webinar in Omics-based tools for biotechnological processes. My name is Anna Maria Salzano. I work in the Proteomics and Mass Spectrometry Laboratory at the Institute for Animal Production System in Mediterranean area in Naples, that is part of the Italian National Research Council. I will talk you today about proteomic strategies to characterize recombinant proteins and to study the response of biotechnology-relevant organisms to perturbations. The research activities at ISPAM CNR are primarily focused on animal production and are based on a systemic approach that considers the animal production system as a whole, moving from plants used as animal feeding to the animal farming up to the product's quality. ISPAM CNR is part of the Italian IBISBA ProTens joint laboratory. This IBISBA node in Naples has been set up to offer scientific and technical expertise for proteomics, interactomics, and peptidomics experiments, to support the research in industrial biotechnology and to improve application in pharmaceutical and agroindustrial processes and in synthetic biology. These technologies can be used to study proteins and peptides present in a biological tissue, in a fluid, or in a cell compartment. And in particular, they are able to describe when and where proteins and peptides are present, the rates of their production or degradation, how proteins are modified, as in the case of post-translational modification, the movement of proteins and peptides from one cellular compartment to another, the involvement of proteins in metabolic pathways and molecular processes, and how proteins interact with other biological macromolecules. Nowadays, proteomics and peptidomics are based on the combination of chromatographic, gel electrophoretic, mass spectrometric techniques, and bioinformatics. At the ProTens node of CNR in Naples, there is a fully equipped and advanced mass spectrometry-based platform for proteomics and peptidomic analysis. This platform is based on robotics for sample preparation, enzymatic digestion and handling of multiplates, a MALDI TOF instrument for rapid profiling of proteins and peptides, two Orbitrap-MSMS instruments equipped with nanoLC chromatographic systems for quantitative analysis of proteins and peptides, and one of these two Orbitrap instruments is also equipped with an ion analyzer, able to perform ETD fragmentation that is specifically employed for determination of post-translational modification like glycosylation and phosphorylation. There is also a dedicated platform for bioinformatics with proprietary software for the database searching of mass spectrometric data, Mascot, Proteome Discoverer, and Bionics, and other several tools for functional analysis of the results. Using the technologies I've just described, we can perform a series of mass spectrometry-based experiments spanning from the identification of peptides and proteins present in a given sample to the quantitative and comparative studies. Quantitative proteomic analyses are focused on the determination of changes in protein levels that follow specific cell treatments or are associated to different conditions. It is also possible to perform a quantitative evaluation of the changes in proteins post-translational modifications. For interactomic studies, it is possible to identify protein partners of specific proteins and also to afford a comparative study to identify protein partners and determine the protein composition of the complex in different cellular conditions and biological contexts. Mass spectrometry can also be used to identify protein and peptide fragments originated by the action of endogenous enzymes. On the one hand, it is possible to define the substrate specificity of a given enzyme or sample that shows proteolytic activity. On the other hand, it is also possible to characterize the degradome of a sample and predict the peptide composition after its proteolytic degradation. There are mainly two general approaches that can be adopted in the mass spectrometry-based proteomic studies, either gel-based or gel-free approaches. In the first one, proteins are extracted from the biological matrix, resolved by bidimensional electrophoresis for protein visualization and quantification. Then the spots are excised, subjected to enzymatic digestion, and then identified by mass spectrometry. For gel-free proteomic analysis, proteins are extracted from the biological matrix, and the whole protein extract is directly subjected to enzymatic digestion. Then the peptide mixture is separated by chromatography and finally is analyzed by mass spectrometry, both for protein identification and quantification. Nowadays, most of the proteomic studies are based on the gel-free approach, in particular for quantitative analysis. In this case, after tryptic hydrolysis of the protein mixture, peptides are eventually labeled using a specific reagent for MS-based protein quantitation, which allows the quantitative comparison of 2 or more samples up to 10 samples in a single experiment. Both in the case of label-based or label-free experiment, an accurate statistical analysis has to be performed on mass spectrometric data by taking into consideration the analysis of biological replicates. Finally, some data interpretation work is required based on functional analysis of the proteins differentially represented in each condition or sample. One possible application of the technologies I've just described is the structural characterization of recombinant proteins, which is an important step for the production of proteins expressed in exogenous host systems, like recombinant antibodies, other therapeutic proteins as enzymes or vaccines. In this case, the verification of the primary sequence of the expressed proteins is necessary to exclude the possible presence of truncated, mutated, and elongated forms of the protein. Dedicated mass spectrometric procedures can also be performed for the verification of the correctness of disulfide bridges that are essential for the proper folding of the protein. Finally, probably the most important issue for the study of recombinant proteins is the characterization of the post-translational modifications, because they are fundamental for the protein's activity and depends on the expression system. For example, I'm going to show you the analysis of protein glycosylation we have carried out in our laboratory on several recombinant antibodies expressed in nail root cultures from Nicotiana bentamiana plants, infected with Agrobacterium rhizogenes, transformed with the plant expression vector. Secondary roots originated in vitro from leaves have been cultivated in sterile conditions, and recombinant proteins have been purified from the culture medium. Several chromatographic steps have been employed for protein purification, and finally a reducing SDS page is performed to purify the protein chain bearing the glycosylation. On the basis of the amino acidic sequence of the recombinant construct, we have chosen the best proteolytic enzyme to get the optimal peptide length for mass spectrometry analysis. Finally, the protein has been digested in gel and the peptide mixture has been analyzed by mass spectrometry. The recombinant antibody expressed in this study is named H10 and is directed against the tumor-associated antigen tenascin C. In this study, we have compared two expression systems, plants of Nicotiana tabacum and glycoengineered plants of Nicotiana bentamiana, in which a downregulation of xylosyl transferase and fucosyl transferase was obtained. The nanoLC-MSMS analysis, which is reported here, allowed us to identify glycopeptides from the two recombinant products and verify that in the glycoengineered expression systems, the glycan moieties does not contain xylose and fucose residues that are typical of plant glycosylation. nanoLC-MSMS analysis allows the estimation of relative abundance of glycoforms in each sample from the ion intensity of the corresponding mass spectrometry signals. In both cases, there is one main glycoform, and in the case of glycoengineered plants, the glycoform corresponds to a typical mammalian glycosylation. Here, there is another study in which the antibody analysis is directed against the CD20 protein based on rituximab, a monoclonal antibody used in the treatment of malignant lymphomas. The recombinant product in the third panel indicated as IC is the same antibody fused with a human interleukin. The analysis reported here is the MALDI-TOF analysis of the glycopeptides enriched by an hydrophilic nanocolumn by using the IRIC resin. Also, in this case, the glycoengineered expression system has produced a protein with mammalian glycosylation. In this study, the same antibody anti-CD20 has been expressed in nail roots using an hormone, the dichlorophenoxyacetic acid, in order to increase the amount of proteins expressed. The glycan structures have been assigned on the basis of measured mass values, fragmentation spectra, and considering the unlinked oligosaccharide structure reported for eukaryotic organisms. The relative abundance has been calculated from the total ion current of the signals associated with each glycopeptide and is referred to the total ion current of all glycopeptides in the sample. With this analysis, also minor components have been identified and quantified. As shown from these studies, mass spectrometry analysis can be used to verify the quality of recombinant products through the characterization of protein glycosylation in order to evaluate both the expression systems and the purification strategies. Another application of proteomics in the field of biotechnology is the study of microorganisms, as for example bacteria used for the production of antibiotics or used as whole organisms, as in the case of probiotics. I'm presenting a collaborative study, which is still going on in our laboratory, on Lactobacillus reuteri. The project is aimed at evaluating the possibility of using this probiotic as a food supplement for astronauts in space missions. And for this purpose, it is necessary to assess the changes undergone by the bacterium in microgravity conditions. The bacteria have been subjected to simulated microgravity in order to define the adaptation of the proteome to these growth conditions. In particular, our collaborators performed experiments using two rotatis devices to simulate microgravity, the rotating wall vessel and the random positioning machine. We have used the gel-free and label-based technology for protein identification and quantification. We analyzed at the same time 6 samples corresponding to the 2 different treatments for microgravity simulation, each one with duration of 7 hours and 15 hours, plus the corresponding 2 control samples. Proteins have been extracted from the bacterial pellets, quantified in order to work on the same amount of material, and then processed. Each sample has been labeled with a different PMT reagent, and finally, samples have been mixed before MS analysis. The MS analysis on the Orbitrap instrument allows the fragmentation of labeled peptides, and the analysis of the MSMS spectra by dedicated software provides both protein identification and quantification. The output of this experiment is a list of proteins differentially represented in the analyzed conditions. The interpretation of proteomics data is then obtained by functional analysis carried out by a plethora of different tools for functional classification. We have only preliminary data up to now. We looked at the protein underexpressed and overexpressed in the 2 microgravity conditions in comparison with the control conditions. We have observed more proteins underexpressed than overexpressed as a consequence of the microgravity condition, both after 7 hours and 15 hours of treatments. It seems that Lactobacillus reuteri exposed to microgravity conditions react by repressing its metabolism, as confirmed by the fact that several proteins involved in DNA replication, transcription, and translation have decreased. Proteomics data will be further investigated to understand how simulated microgravity could alter some physiological characteristics of Lactobacillus reuteri, in particular with regard to tolerance toward the stress condition encountered on space missions. In conclusion, IBISBA offers a platform for proteomics, interactomics, and peptidomics studies based on advanced chromatographic and mass spectrometric technologies combined with bioinformatic analysis. The potential of the technologies at our disposal allows the analysis of single purified proteins and peptides, as well as the analysis of entire proteomes, interactomes, and peptidomes. Label-based and label-free strategies based on mass spectrometric analysis that we use currently in our laboratory allow the comparative and quantitative evaluation of numerous samples at the same time. And finally, dedicated experiments can also be performed by MALDI-TOF-MS profiling and nanoLC-MSMS sequencing for the quantitative analysis of post-translational modification. Thank you for listening, and if you have any question, I'm here to answer.